Well, thank you very much for the opportunity to talk to you here today, and thanks to the Cardinal and the Swedish Embassy and the Vatican, as well as the organisers of this meeting, EACR. I'd like to talk to you about a strategy that we've taken in Australia to try and implement at a national scale what I believe is a new model for cancer care. It's sometimes difficult to get a sense of perspective about these things, but back in the 1990s, which is when I did my training, we didn't use a molecular or scientific concept to design treatments for cancer patients at all. It was only just beginning to emerge with imatinib and Brian Druker's paper, and that expanded through multiple indications after that time point. But today, I'd have to say, if you look at lung cancer alone, the landscape has completely transformed. That's driven largely out of industry, in my view. And if you look at single-payer systems which are trying to distribute taxpayer dollars, you'd have to say that their adaptation has been vastly slower than the trillions of dollars which are being invested by industry in that model of care. The question is, how do we bring those two worlds together, in my view? And in fact, I think what we're seeing is that science works. If COVID didn't prove that, and HIV didn't prove that, and polio vaccine didn't prove it, it's certainly proven by, I think, what's happening in cancer, but it's not being perceived in the same way, and I think that's the challenge for our community today. I need to disclose some of my conflicts of interest. I'm the Chief Executive Officer, unexpectedly, of a company, a non-profit company, which is designed precisely as a social solution to the challenges of delivering precision medicine. And we work very closely with a broad range of them, the companies that produce the drugs that we need to treat our patients. I am actually a medical oncologist. I specialize in rare cancers, sarcomas actually. I'm a clinician scientist, and I did not expect to be a CEO. But there you are. So this is the stats in Australia. In 2022, it's estimated there'll be about 160,000 diagnoses of cancer and about 48,000 cancer deaths. The 5-year survival is really very good at 70%. But the interesting thing is this enormous number of patients who are alive today with a diagnosis of cancer. Cancer in Australia costs more than $10 billion a year. It was estimated to be $10.1 billion in 2015-16. That will have risen. It's something like 11 or 12% of GDP. And a factor which we also have to bear in mind, money keeps on coming up. Health has an inexorable demand on budgets, on GDP, gross domestic products. And in every developed country around the world, it's been rising at around 0.5 to 1% per year for the past 20 years. Such that in the US it's about 17%, and in Australia it's 12%. We can see the future geographically located overseas. That's what we have to address, along with aging populations and a diminishing taxpayer base. I want to talk about rare cancers, because they were raised several times. It's somebody commented on the fact that all cancers are becoming rare, but histologically rare by the previous classification that's developed in the 19th century about histology and anatomy, account for 42% of all cancer deaths due to rare and less common cancers. And that's caused a problem in the empiric era of drug development, because we needed to use the classification of histology and anatomy to design our breast cancer trials, our bowel cancer trials, and so forth, which left this long tail, again, this long tail concept of individually small cancer types typically left behind in terms of access to the benefits of research. And as a consequence, although they're 30% of cancer incidence, they're actually 42% of all cancer deaths. And this is an area where one can make a positive statement about science and its ability to democratize access of cancer patients to new treatments. We had to set up Omico in order to unify a state-based health system in Australia. And we had to set up a company because standard medical research funding with a project-based endpoint over a finite timescale wasn't going to cut it when you're talking about a model of care which we clearly all envision to be something that's going to be around in perpetuity. And in setting up a company, we're able to bypass state and federal government systems to unify cancer centers throughout the 8 states and territories across the country, the states and territories being responsible for high-tech care in hospitals. And the only way to do that without going through the governments was to set up a company, a non-profit company called Omico. And it has a broad remit. I'm going to talk to you mostly about molecular screening and therapeutics, because that's the area where we can see a sustainable business model going forward for implementation of precision medicine, but actually very interested in personalized risk management, and particularly in the idea of secondary cancer early detection strategies. For example, if a 30-year-old were to have a myocardial infarction today, we would never dream of curing them of their heart attack and sending them back into the community without any further personalized health maintenance plan. But that's exactly what we do for the most part with people who get diagnosed with cancer at a young age. And the idea that one might be able to do the same thing as cardiovascular disease, manage the risk factors for a disease in order to prevent the disaster, hypertension, hyperlipidemia, and so forth, have been used very effectively as stratifying factors which have allowed us to prevent people from getting heart attacks at an age in the 50s, such that cancer now has overtaken cardiovascular disease as a leading cause of death in higher income countries. The decline from the management of risk in cardiovascular disease is 60% over the past 20 or 30 years. That's what we should be aiming for. So I'm not going to talk about that, but I think it's an incredibly important area, and genomics and genetics has a lot to teach us about a disease which is fundamentally genetic. The health system reform is absolutely critical to what I'm going to talk about later. We need to make this a mainstream model of care rather than an esoteric area of research run by medical research budgets rather than health services budgets, or even a broader access to support from finance and treasury and seen as a legitimate part of the economy. And that's because we typically regard, and I keep on hearing messages about the separation of industry. In fact, only one person in this meeting has talked about industry, though they produce 95% of drugs. And that was Edith, who mentioned private-public partnerships. We need to rethink how we interact with industry in drug development in particular, because it is simply not sustainable. And of course, at the center of everything have to be patients, because genomics allows us to study humans in their native state, which is by far and away the best way of identifying the pathways that we need to tackle, because humans are the disease, where the diseases occur. And we now have the tools to understand the genetics and the biological pathways that human patients are prone to. So we set up, using this company, a national program which started off with 8 centers and federal government funding of roughly $50 million. Within 3 years, from 2019 onwards, we're now at 23 centers, and we've just opened in New Zealand. And this is because of the appetite of the clinical and the patient communities to drive integration of molecular screening and therapeutics program into their clinics. These are data on recruitment. We expected to reach 3,095 patients by the end of our 5-year term, which is in 2020, beginning of 24. We're now at 7,000 patients 1 year out, and that's purely due to demand. More than 80% of the 500 practicing medical oncologists in Australia have referred their advanced cancer patients onto this program to get molecular testing, because it isn't otherwise available outside of medical research, at least with any equity. And the interesting thing is about 82% of the patients referred are rare and less common, which is exactly what we'd hoped we'd see. We'd see areas where with the greatest burden of mortality and morbidity from cancer, and those are pancreatic cancer is self-evident. Amongst the top 8 cancers that we see, cancer of unknown primary, ovarian cancer, lung cancer, pancreatic cancer, brain cancers, and my own specialty of sarcomas. I was pleased to see patients finally getting access to some form of personalized therapy. And of course, the rich molecular landscape that's been identified, I showed this not because I'm asking you to look at it in any detail, except to say that up the top, you'll see that tumor mutational burden is up there. And immunotherapies have, as somebody commented earlier, transformed clinical care. And tumor mutational burden is regarded as a predictor of response to immunotherapy. Now, that's across all of these 82% of rare cancer types. We see a distribution of high tumor mutational burden, which predicts response. But we need to develop trials which allow those patients with rare and less common cancers who have tumor mutational burden to get screened, at least in single-payer systems, so that the regulatory bodies will accept both testing and treatment for those patients in perpetuity. And clearly, treatment's the key here. The genomics is fantastic, and it's really, from a scientific perspective, an absolutely incredible area to study, like a terra nullius, to use an Australian expression, a completely it's an unexplored landscape from a scientific perspective, which is only just beginning to be touched upon. But the treatments are what we wanted to focus on. And in the past few years, we've ended up supporting 23 clinical trials, what we've developed in a basket trial design, so that we have patients, somewhere to put the patients once we identify a druggable target. And almost 900 patients in the past 4 years have gone on to trials as a consequence of the screening program to date. Overall, about 2/3 of patients have a druggable recommendation. And this is a population of advanced cancer patients of any histotype with a performance status of 0 to 1 that would be fit for therapy, there's absolutely no point in science providing hope to patients who will never benefit from treatment, and that's bad medicine. So we're focusing on those patients who who have run out of conventional options and have no place to go. 2/3 of those have a druggable target, and you see the different nomenclature there, C1, C2, and C3. That refers to the different types of clinical trials that we refer the patients onto, because that's the majority of our strategy for getting patients treated. And about 50% of the patients that we make a recommendation on actually go on to get onto trials. The other 50% are probably a combination of distance, lack of access, and becoming too sick, to be frank, to go on to a trial. Now, we've developed a classification system in our reports that's intended to guide clinicians about the strength of evidence for the recommendation. And that's outlined here. There are multiple of these sort of classifications that are around the world. The top tier, tiers 1 to 3, represent drugs which have attained such a degree of evidence of benefit that they've been approved in Australia by our Therapeutic Goods Administration and or reimbursed. Tier 2 is those that are approved by the FDA, for example, or the EMA, but not approved in Australia, with positive phase 3 data. And Tier 3 data based around positive phase 2 trials or better. And then there's a whole series of experimental treatments, including an important category where we repurpose drugs. This often happens with rare cancer patients. We see a target, we know it works in one disease, and we transfer it to that disease on the expectation of benefit. And that's an appealing idea, but there's reasons, as I'll show you, to doubt whether that's that can be sustained outside of a clinical trial. These are our outcome data. I was very pleased to see that 37.5% of the 3,800 odd patients in this particular analysis had a tier 1 to 3 biomarker. And if you look at the graph to the right, the curve that is most to the right represents tier 1 to 3 biomarker and the extension of survival in that patient population, by comparison with an equivalent population who received non-matched therapies. So they received treatment. These aren't just 2 sick patients in the control arm. This is the impact of doing molecular screening and then access to cognate therapies overall. And you can see the general benefit there is highly significant statistically. It also applies to it sort of applies to tier 4, but it fails to reach its marginal and statistical significance. But tier 3B, the repurposing, definitely did not show a clear enough benefit in our population to make it obvious that one should be extrapolating from one cancer type to another. And that we've known from BRAF and melanoma and colorectal cancer for some time. You can't be safe in assuming it doesn't work, because we do know things like TMB do work across cancer types, but it definitely is not safe to disregard histotype in choosing a molecular targeted therapy. This is for rare cancer population. So these patients, 82% of the cohort, show exactly the same benefit for tier 1 to 3 treatments and show the same lack of benefit when repurposed therapies are being applied. Interestingly, tier 4, which is based on preclinical evidence, shows some promise in this end-stage cancer patient population, which suggests that emerging out of basic research, the Bayesian prior of knowledge of biology that drives drug development, rational drug development, does work to increase the probability of benefit for cancer patients. And this is one of the reasons why I think, basically, no cancer patient should go to their grave without having access to the current model of rational drug development. It's increased in its probability of objective response 6-fold compared to the 1990s, because we now have a biological framework within which to design drugs to appropriate targets. And this means something for patients. So this is a patient with pancreatic cancer who enrolled on the program and turned up an NRG1 fusion. This is a rare, recently described fusion for which there are drugs just beginning to be developed. And this particular patient had to get access to the drug prior to us being able to open the trial. And pancreatic cancer is a dreadful disease, especially when it becomes refractory to conventional treatments. But this patient, being exposed to a HER3 inhibitor, showed a response both objectively and in terms of biomarkers. And that's not something you expect, and it's one of the reasons why one should be considering this even in situations where you think the expectation of response will be low. Now, pancreatic cancer is also useful because it illustrates the long tail principle. Things like NRG1 fusions are extremely rare, but the cumulative number of rare targetable events in pancreatic cancer is now better than 10%. And for a disease which has a 10% 5-year survival, and that's doubled from 6% only 10 years ago, this is a really important area. And with G12D inhibitors coming along, KRAS G12D is going to make a big difference in this population. Lung cancer is transformed. Cholangiocarcinoma is about to be transformed. There's no reason why this logical process ought not to extend as we explore new and less common indications. And I want to talk about pan-cancer biomarkers because that's the only way we're going to design appropriate randomized studies in rare cancers. We're going to have to combine them and use a molecular biomarker that we use to select patients for treatment. And you see TMB on this particular analysis shows that TMB does predict response to targeted therapies, particularly immunotherapies. It's the bottom part of this forest spot. The bottom line is high tumor mutational burden. But just above that is a new biomarker we identified through molecular analysis of patient populations coming onto the program, and those are mutations in the SWI/SNF complex. And for those of us who spend a lot of time looking at molecular data of genomics of cancer patients, it is an extremely common type of mutation. And if that turned out to be true, I could imagine a randomized study where we put patients in aggregate with rare cancers, say less than 6 per 100,000, onto a randomized study to generate the same rigor of data. It just requires a change in mindset from the 19th century classification to one that addresses the inequity of access that rare cancer patients have had for cancer over quite a long period of time. And I'm going to stop there for the sake. Thank you.